Alright, this video is your pre-lab for cellular respiration lab, the observation, which is part of your pre-lab. Temperature affects cell respiration in yeast. The question, how does ambient temperature affect the respiration rate of yeast? That is a question. Okay? So your hypothesis is the answer to the question, and then of course you have a prediction, which is an if-then statement. Remember, when you're doing your prediction, the if part is referring to what are you doing, and then the then part is what are you trying to determine. Okay, so that should help you in your prediction. So your observation, question, hypothesis, and prediction needs to be completed before you do the lab. If you do not have it completed, then you cannot do the lab. So let's talk about how we're determining if ambient temperature affects respiration rate or not. And we have this diagram here. So you're going to have a test tube. Inside the test tube is going to be water, and then you're also going to have a tube, or like a bulb of a pipette. Inside that bulb, you're going to have some yeast and some sucrose, which is sugar. So you'll have some sugar water in with the yeast. Okay, so sucrose is made of two sugars. Among them is glucose and fructose. So the glucose is going to, and the fructose, will both be broken down through cellular respiration, and the yeast is going to produce carbon dioxide bubbles. Okay, so those carbon dioxide bubbles are then going to accumulate inside the tube and right about here. Okay, and they will come out the top, and you'll be able to count them in the water. So how we're going to affect, or how we're going to change the temperature is mainly putting in different temperature water. Okay, so what you'll do is I will put this whole thing, okay, this test tube in a, not this test tube, but the pipette. You'll remove that, and then you'll put in different temperature water. And then when you put the tube back in, now this yeast and sucrose will be exposed to those different temperature water, and you are going to look at three different temperatures. You're going to look at a warm temperature, you're going to look at a room temperature, and you're going to look at a cool temperature. Now the room temperature is going to be your control, so make sure you include that in your lab report. Okay, you do have a control group for here, or for this lab. Okay, warm, make sure you get the temperatures according to the thermometers. Okay, I will let you know what the room temperature water is, so you don't have to worry about getting that temperature. All right. So you're going to count bubbles for 5 minutes at each temperature for a grand total of 15 minutes. Your data table will look like this. Okay, you want to get the total number of bubbles at warm temperature, at room temperature, and at pool temperature. So you get the total number of bubbles here, and then, so let's go ahead and say that this is X, Y, and Z. Okay, and in order to get this answer over here for bubbles per minute, it would be X divided by 5, this would be Y divided by 5, and of course Z divided by 5, and that will give you bubbles per minute. Okay, so you're definitely going to want to have your data table filled out as well. You'll need to get that done before you come to the lab. Your lab will also have a graph, and this should look very similar to your photosynthesis lab. On the y-axis, we're going to have bubbles per minute. On the x-axis, we're going to have temperature. You will do another best fit line. So, for example, if you were to get this dot, this dot, this dot, this dot, and this dot, okay, you can tell that this has a trend line going in that general direction. Okay, I'm not saying that this is your graph or not, but you will need to plot your dots and then do a best fit line. Okay, so a little refresher. What you need in order to do the lab, you need to have your entire right page completed, okay, for your pre-lab stuff, which is your observation through your prediction. You do not need to worry about your procedure yet, or your variables, or your control group, even though you should be able to figure out what those are. Okay, I'm not requiring those to be done before you come into the class. And then you also need your data table, which is on the left page. Okay, so your data table and your observation, question, hypothesis, and prediction need to be done.